Hello, in this video I will give a short recap of lysosomal storage diseases as told by myself, Mark Mitzstein. So first of all, what is a lysosome? So a lysosome is generally thought of as a disposal or recycling organelle of the cell. And the lysosome works by taking in complex biopolymers such as polysaccharides, proteins, and nucleic acids, and then converting it into monomers such as monosaccharides, amino acids, and nucleotides, and spitting them out for the cell to use for other metabolic or catabolic processes. And the way that these polymers are broken down is through the action of a series of enzymes called acid hydrolases. So these are called acid hydrolases first because they work in a low pH environment, which is what the inside of a lysosome is like. And secondly, they're called hydrolases because they take the activity of water to split up bonds within biopolymers to release the monomers, as shown over here. Now, to get some idea about how lysosome and lysosomal storage diseases work, we're going to zoom in a small portion of the cell to look at membrane trafficking pathways that are involved in lysosome biogenesis and where they get their substrate for disposal. So the lysosome receives input from two major pathways. First of all, the lysosome get most of their cargo, i.e. what they're going to degrade, from endocytic pathways that start at the cell surface and lead to a series of membrane-bound compartments which eventually fuse with the lysosome to deliver cargo consisting of surface proteins, surface membrane, as well as extracellular ligands which are bound to the surface. Meanwhile, the enzymes found in the lysosome are manufactured in the ER and Golgi apparatus, and then they go through their own trafficking pathway directly to the lysosome, where they are involved in degradation of products. There is actually a second pathway that the lysosome can get components for degradation, and this is an intracellular pathway, and it works through the autophagy mechanism. So in autophagy, which I'm not going to go into details of, but if there are components of the cytosol, including damaged organelles and other things which the cytosol wants to get rid of, a membrane forms or in the cytoplasm, enclosing organelles and other macromolecules, forming what's known as an autophagosome. And then the autophagosome then goes on to fuse with the lysosome, thus delivering internal cell membrane components for degradation within the lysosome. So what are lysosomal storage diseases? Well, in a nutshell, what they are loss of function in one of the 50 odd genes, including one of the lysosomal hydrolases. And the consequence of lacking one of these hydrolases in the lysosome is that the macromolecule that was going to be broken down by that enzyme is no longer broken down. The macromolecule then accumulates inside the lysosome, causing lysosome dysfunction and eventually death or dysfunction of the whole cell. Now, while this mechanism is relatively straightforward, it actually can lead to very diverse diseases depending on which particular enzyme is missing, and it's mostly because of different cell types have different requirements to degrade particular types of macromolecule. Lysosomal storage diseases are divided into three broad classes. There's the so-called sphingolipidoses, the mucopolysaccharidoses, and then there's one form of a glycogen storage disease. So first, let's talk about the sphingolipidoses. So sphingolipids are a series of complex fatty acid-derived molecules. There are a number of them, and they're all based on this common tail of a chemical called ceramide. So ceramide tail is shown here for all of these different sphingolipids. And then there's modification of the head group of the ceramide to make the whole series of different sphingolipids you see here, including ceramides, sphingomyelins, cerebrocytes, and gangliosides. Now, it's important that you do not have to know the structure of any of these sphingolipids. You just have to know that they exist and they have a broad idea of what their structures are, which is a lipid tail with a modified head group. The names give you some clue sometimes about where they're from. So, for instance, sphingomyelins are very abundant in myelin lining nerves, and gangliosides here over on the right are quite often in ganglia, i.e., nervous system. Now, the sphingolipids are broken down within lysosomes in a series of enzymatic steps. So here is a little graph here. And so basically, these are a series of steps which remove the side groups from the different sphingolipids and eventually release the ceramide central lipid. And with each of these breakdown steps is associated with a specific enzyme. So for instance, the breakdown of GM2 gangliosides to something called GM3 gangliosides is catalyzed by this enzyme hexoaminidase, while the breakdown of, say, sphingomyelin is broken down by an enzyme called sphingomyelinase. And to cut the story very short, it is defects in each of these enzymes which underlies lipid storage diseases. So nearly all of these enzymes is associated with a well-known lysosomal storage disease. So for instance, the breakdown of GM2 gangliosides to GM3 gangliosides, if that enzyme hexaminidase is defective, what you get is Tay-Sachs disease. And similarly, you can see for all these different steps, there are different diseases associated with it. And here is a table, this is derived from first aid, showing which the enzyme deficiency is over here on the left, what substance is accumulated when that enzyme is deficient, what the principal signs and symptoms are when someone suffers from one of these diseases, and the commonly known name of the disease. And some of these are clearly quite famous diseases like Tay-Sachs disease or name-and-pick disease. Now, this is one of the few examples in medical school in which I'm going to recommend simply memorizing these tables. So there's a good table, as you see, in first aid. There's good tables online. There's many videos which gives you ways of memorizing this. And this is just required to answer any quiz or step one questions to do with these different diseases. So the next class of lysosomal storage disease I want to talk about are called the mucopolysaccharidoses, and these are failures to break down chemicals called glycosaminoglycans, or GAGs. So GAGs are large complexes of negatively charged heteropolysaccharide chains, so not lipids in this case, but polysaccharides, and they're often associated with some core protein that makes something called proteoglycans. And the part of these proteoglycans is these are extracellular matrix molecules that can bind a lot of water and make gels, and they're used in parts of the body that need like lubrication, like joints, or large volumes of gel-like liquid, like inside of your eyeball. So when the lysosome is unable to break down these glycosaminoglycans, GAGs, and there's two main enzyme deficiencies shown over here on the left. You accumulate certain GAGs in here inside the lysosome, and that leads to certain symptoms within the patient. And then, for some reason that's just not really understood, the patients suffering from mucopolysaccharidoses end up with these very, very distinct facial features. So over here on the right, you can see a young boy with what's called Hurler syndrome, and you can see these like bulging eye sockets, funny shaped mouth, funny overall head shape. As well as this, these patients also have developmental delay. They get cloudy corneas, heart valve problems, hepatosplenomegaly, and this disease tends to be fatal within 3 to 4 years. So next, I want to talk about an interesting subclass of the mucopolysaccharidoses called eye cell disease. So eye cell disease, and there is a patient here on the left who has eye cell disease, and I want you to compare the face of this patient with the boy you saw in the previous slide who has Hurler syndrome. And what you can see is that these patients share a number of the unusual facial features, such as the heavy eyelids and the strange shaped mouth. And as well as these facial features, these patients also share abnormal skeletal development, they have restricted joint movements, corneal clouding, and also severe intellectual disabilities. And so, because of this, eye cell disease is also known as mucolipidoses type 2, as is written here. However, despite the similarity in outward appearance, eye cell disease has a number of unique clinical characteristics. So one thing that if you look at the leukocytes of people with eye cell disease, but not with other mucodoses, what you'll see in eye cell disease is the leukocytes fill up with these inclusions, large inclusions throughout their cytoplasm, shown white here in this EM stain. And these are actually these inclusions are the source of the disease name, eye cell disease or inclusion body disease. So how does eye cell disease work? Well, what we're going to do is go back to a slide we saw earlier when we talked about the trafficking pathways that get components of the components and enzymes to lysosomes. And what we talked about then was how lysosomes get a lot of the things they have to degrade from the cell surface, as from over here, this pathway. And then, more importantly for this discussion, they get the enzymes they're going to use from this unique pathway which comes from the Golgi apparatus. How do these enzymes go from the Golgi apparatus to the lysosome? So it turns out that the hydrolytic enzymes, which are made in the Golgi and then go into the lysosome, are glycoproteins, meaning that they're modified on some amino acids by sugar side chains. And one particular sugar, which is always found in the side chains of these hydrolytic enzymes, is the sugar mannose, shown here by this green dot. And mannose structure is not important to know, but it's a typical hexose sugar. So it turns out that mannose plays a particularly important role in trafficking of these enzymes because mannose on these side chains can undergo a phosphorylation event catalyzed by the enzyme called N-acetylglucosamine 1 phosphate transferase, which attaches a phosphate group onto this mannose residues. And then inside the Golgi apparatus, there is a particular receptor called the mannose 6-phosphoreceptor, which recognizes these phosphorylated mannose residues and makes that particular protein then go away from the trafficking pathway which goes to the cell surface, but instead traffics to the lysosome. So phosphorylated mannose is a signal for trafficking to the lysosome. So what you might not be too surprised to hear is that the defect in eye cell disease is a defect in the phosphorylation of this mannose. So the defect in eye cell disease is a mutation in this GNPTAB gene. And so that mannose never gets phosphorylated to here, thus the hydrolytic enzymes are never recognized by this receptor, and thus it's never trafficked to the lysosome. Instead, the hydrolytic enzymes follow an alternative default pathway, and instead of going to the lysosomes, they're trafficked actually to the cell surface and out into the plasma. Because of this, eye cell disease has another distinctive feature which distinguishes it from the other lysosomal storage disease, in that you find a high level of lysosomal enzymes in the blood because they've come out by this default trafficking pathway. And so what are actually all these inclusions you see in in the cells here? Well, these are actually really packed lysosomes because these lysosomes cannot degrade essentially any of their cellular components. They're not just missing one enzyme like a normal lysosomal storage disease, but they're missing all their enzymes. So they form these large inclusion bodies within the lysosome. And then these two features, one, the inclusion bodies within the lysosomes, and also the high level of lysosomal enzymes in the blood, are the things that differentiate eye cell disease from Hurler syndrome, which it superficially looks like. I just want to point out that eye cell disease, despite having a trafficking problem of all lysosomal hydrolytic enzymes, does not overlap with all the lysosomal storage diseases. For instance, the mental decline seen in Tay-Sachs disease or the bone problems seen in other diseases. And this is not really understood why, but it suggests there's enough of the enzymes that are involved in these other diseases making it to the lysosome to carry out the functions that they're required, but not enough of the polysaccharide digesting enzymes to avoid getting eye cell disease, to avoid getting a mucodosis. So finally, I'm going to talk about one form of glycogen storage diseases. So there's actually quite a few glycogen storage diseases. This one's called type 2, also called Pompe disease. And most glycogen storage diseases have nothing to do with lysosomes at all. So glycogen is a polymerized form of glucose, and it's not normally found in lysosomes. It's found in the cytoplasm of cells. And there are plenty of enzymes within cytoplasms that both build up and break down glycogen. And so how does this manifest as a glycogen storage disease? So first, the defect in Pompe disease is indeed in a lysosomal enzyme. This enzyme is called alpha-1,4-glucosidase, also called acid maltase. Maltase is just a name for an oligomer of glucose molecules, like a glucose molecule is called maltose. And so glycogen is actually a form of elaborate maltose. And what this lysosomal enzyme does is it catalyzes the conversion of glycogen to glucose within the lysosome, so not within the cytoplasm. So what happens if you lose this enzyme? You result in glycogen accumulation in the lysosome, and this can be seen in the staining by all these white dots that you see within muscle cells here, because the lysosomes are now packed with glycogen and are starting to fill up the muscle cells. So the symptoms of glycogen storage disease type 2 is mainly based on which cells have the most glycogen, and that is primarily muscle cells. And so the symptoms you see of a patient suffering from glycogen storage disease is that you'll see cardiomegaly, the heart cells will build up, hypotonia, weakness of the muscles, eventually cardiomyopathy and difficulty breathing, muscle weakness, and you also get hepatomegaly from the extra storage of glycogen within the liver. And these are symptoms that are due to tissue damage caused by accumulating glycogen within primarily muscles. So there's one question this doesn't answer is where do lysosomes get glycogen from? So I already told you that lysosomes mostly get their components from trafficking from the cell surface, as shown here. But glycogen doesn't come from the cell surface. It's a cytoplasmic component. So where it actually comes from is from this other process which I mentioned earlier. It's from autophagy. So as I talked about earlier, the autophagic process is when a membrane gets built within the cytoplasm to surround organelles and other components you want to get rid of. Now, the muscle cell is full of glycogen, as denoted by these red dots in this diagram. And what happens is a certain amount of this glycogen ends up getting trapped within the autophagosomes. And so when the autophagosomes eventually go into the lysosome, that glycogen comes along with them. And in the absence of this particular enzyme, acid maltase, they can never get rid of this excess glycogen from within the lysosome, and the lysosome eventually malfunctions and leads to cell destruction. So with that, that is my brief guide to lysosomal storage diseases, and we will discuss these in class next week.